Hi there, my name's Ushin, and our group chose to do our project on CRISPR-Cas9, which is a novel technology which has been a great source of excitement in recent years, and it could hold the key to treating a wide variety of illnesses. So what is CRISPR-Cas9? It's a groundbreaking development that could revolutionize the field of biochemistry. So the technology is capable of correcting dangerous genetic disorders, and it opens the door to treatment for illnesses that have previously been understood to have been incurable. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and was first discovered by Francisco Mojica in the University of Alicante in Spain in 1993. And the sequences are observed in prokaryotes and are basic bacteria's way of defense against viruses. So whereas we have an innate immune system, they have CRISPR. CRISPR-associated protein 9, or Cas9, is the second key component of the system, and it was discovered by Alexander Bollatin in 2005. And Cas9 functions to cleave two strands of foreign DNA it identifies, such as that of an invading species, and it allows sequences to be added or removed as necessary. So when this system of CRISPR and Cas9 are coupled together, an adaptive DNA repair system has been hypothesized as a really promising possibility. The system is efficient and can change small sequences, editing a problematic gene in ways that previous techniques weren't able to. And with other technologies, editing of a gene would have required re-engineering of entire proteins. So the development of CRISPR has the possibility of being a real game changer in our fight against illness and disease. And Madison will go into more detail about the mechanism of CRISPR-Cas9 and that system later on, but an important aspect of the system is restriction enzymes. So I'll hand you over to Matthew, who will go into a bit more detail on these and their importance in CRISPR.
Genetic editing has been around in some form for thousands of years. Early growth of agriculture allowed humans to select for desired genes found in nature through artificial selection. However, it wasn't until 1969 that we began to see a window into molecular genetic editing. The discovery of restriction enzymes in E. coli by Werner Arber and Stuart Lin proved the ability of some cells to edit their own DNA. This discovery showed that these endonucleases could create double-stranded breaks in DNA at or near specific restriction sites. These restriction sites were short, often palindromic repeats found throughout the genome. This discovery made way for further advancements in the early 1970s. By 1972, Paul Berg was able to use a variety of restriction enzymes and ligases in combination to create the first recombinant virus. This was done by combining DNA from a monkey into the SV40 virus. Only a year later, Cohen and Boyer were able to conduct a similar experiment. Only this time, they were able to produce recombinant genes in bacterial cells. This allowed for propagation of genetic material through the following generations of cells. The combination of restriction enzymes and ligases in vitro became the standard for molecular cloning and genetic editing. Over the next few decades, hundreds of restriction enzymes were discovered to improve the accuracy of genetic editing. While this method was often the most effective at the time, it was not particularly efficient or predictable. Overall, these methods for genetic editing were costworthy and unreliable. This led many researchers to search out an alternative method, eventually leading them to the discovery of the CRISPR-Cas9 system for genetic editing.
Within a DNA sequence, there are genomes which encode a series of messages and instructions. CRISPR-Cas9 in general breaks down the DNA and tricks a cell's natural DNA repair mechanism to change these messages the genome contains. There are many Cas proteins that can be used, and they are distinguished by their structure and function. However, the CRISPR-Cas9 system is relatively simple and is the most studied. The mechanism has three distinct parts, which are recognition, cleavage, and repair, and involve two essential components, one which guide the RNA to match a desired target gene, and the Cas9 protein, which is an endonuclease that causes the double-stranded DNA to break, allowing for the modification to occur. Starting with the designed single guide RNA, which is specified to the gene, it recognizes the target sequence in the gene of interest all through its 5' CRRNA complementary base pair component. The Cas9 protein can be thought of like scissors that cut the double-stranded DNA at the site of the 3 base pair upstream to PAM. PAM is short for protospacer adjacent motif and is a short DNA sequence that is 2 to 3 base pairs in length that follow the DNA region that is targeted for cleavage. Each Cas protein has a specific PAM sequence that it is able to recognize, such as Cas9 protein recognizes the sequence 5' NGG3', where N can be any nucleotide base. Once the Cas9 protein is at an appropriate site in the PAM sequence, it triggers the local DNA melting that forms an RNA-DNA hybrid. This allows the Cas9 protein to be activated for DNA cleavage. The Cas9 protein has two nucleases that cleave the complementary and non-complementary base pair strand to produce blunt-ended double-stranded break DNA strands. When this happens, the cell tries to repair the cut with host cell machinery. The repair process is error-prone, leading to the mutation that effectively disables the gene or adds.
Over the last 15 years, CRISPR-Cas9 technology has developed rapidly as the leading method for reliable genetic editing. Many modifications have been made to the experimental procedure over the years for a variety of reasons. Different enzymes, RNA templates, and delivery methods have been employed to accommodate a large amount of tasks. Regardless, most CRISPR-Cas9 systems follow a similar outline. First, a single guide RNA is selected. This is a piece of RNA that contains a guide for Cas9, and a tracer sequence is added to it to allow for easy analysis. Typically, these RNAs are designed synthetically using a virtual design software. This allows researchers to design an RNA guide that will produce minimal negative interactions with surrounding genes. Second, the RNA is annealed to the active enzyme. In most cases, this is Cas9, but other enzymes have been designed to serve a similar function. Once the sgRNA is bound to Cas9, it creates a ribonucleoprotein, aka an RNP. The RNP contains all active proteins and genetic material for the reaction. Third, the CRISPR components are delivered to the cell. This can be done in a variety of ways. A viral method, such as a viral vector, a non-viral method, such as lipid-bound particles, or a physical method, such as a microinjection. Viral methods are typically the most common, but each has their own individual advantages and disadvantages depending on the cell. Finally, after a few days for cellular development, results are analyzed. Many methods exist for genetic sequencing, but methods such as next-generation sequencing and tide analysis have become popular due to their ability to accommodate large amounts of genetic information simultaneously. The entire reaction is a multi-step process requiring lots of equipment, time, and patience.
CRISPR has undoubtedly opened the door to amazing opportunities for the advancement of the human genome. For example, clinical trials are already ongoing, aiming at treating HIV, as is laid out in Ishii et al.'s 2016 paper on somatic gene editing. However, we have to remember that there are risks and limitations that go along with this novel system. Genome editing performed using CRISPR could lead to irreversible damage to patients if incorrectly used. As Chadwick et al. Mentioned in their paper on the possibilities of CRISPR, the system can cause off-target mutagenesis, which can lead to gene disruption and the development of cancers, as well as cell death. And once issues like this arise, it will be difficult to reverse the effects with current technology, unlike with most medications that we use to treat illness with today. There's also the moral issues that follow the possible implementation of the novel system, and as Eduardo Rodriguez laid out in good detail in his 2016 paper. Assuming any possible risks described earlier, such as cancer and cell death, are solved, CRISPR could still represent a big issue to society in a moral sense. Such a technology would only be available to the wealthy and the privileged, and if misused, it could exacerbate inequality throughout the world. Although this technology can be used to cure life-threatening illnesses, it can also be used to enhance desirable genes, leaving the door open to abuse. So if a select few have access to this technology and can alter their genome to become superior, smarter, stronger, healthier, then CRISPR could become more trouble than it's worth. It can't be understated how important it is to make sure that this technology is used responsibly by those who control it and used only to make necessary corrections to the genome. Currently, incurable diseases such as Huntington's would be examples of beneficial targets for CRISPR technology. Such diseases could basically be stamped out through genome editing using this technology, and other diseases of the same ilk would equally be fair game as far as I'm concerned. The use of CRISPR for cosmetic alterations is where the issue is, and it should be avoided. And though this will limit its use, I think it's a necessary boundary to prevent possible misuse of this powerful new system. And now I'll hand you back over to Madison, and she'll go into a little bit more detail on the applications of CRISPR now and in the future.
CRISPR-Cas9 technology has provided many opportunities within the means of precise genome editing. Since first conceived, it has been successfully used in the fields of medicine, agriculture, and biotechnology for a multitude of diverse applications. Other previous genome editing methods have been surpassed, such as ZFN and TALEN, due to its advantages in simplicity, low cost, specificity, and ease of design. Specific interest has been taken about the application of CRISPR-Cas9's ability in the prevention and treatment of human diseases. Research and clinical trials have been employed for a wide variety of diseases, including single-gene disorders such as cystic fibrosis, hemophilia, and sickle cell disease. Further research must be employed to discover the prevention of more complex diseases such as cancer, heart disease, mental illness, and HIV. Further research could also be done to design new grains to improve their nutritional value.